Hello, and a very warm welcome to the AfOx Insaka, the very first one this year. And we are delighted to welcome our guest today, who will be speaking on two topics that could be categorized as first things first: community engagement and food security. What are we as researchers without community engagement? In fact, what is research without engagement? And with the situation we all know too well, I would think nobody needs much convincing. The first talk will tell us about research engagement in global health emergencies. And the second talk, a very popular topic with AfOx, is on research capacity. And this time, not in health, but in plant sciences, and specifically crop genetics. Africa does indeed need a green revolution. I will not stand in the way any longer between you and our speakers today, Noni Mumba and Oluwaseyi Shorinola. Noni Mumba is Head of Community Engagement at Kemri Wellcome Trust, Kilifi. She's an expert at developing best practice strategies for engaging local communities and the public in research. Olu Shorinola is a crop geneticist and holds a FLAIR Fellowship, a very prestigious African Academy of Sciences and Royal Society Fellowship. He's based at Beka Ilri, the Biosciences Hub in East Africa, and was previously a PhD and postdoctoral researcher at the John Innes Center in Norfolk. As usual, the speakers will each give 15 minutes talks in succession, and we will have a Q&A session after both talks. So please do remember to post your questions in the chat box as we go along. And first up is Noni, and her talk is titled Engagement During Global Health Emergencies, Challenges and Opportunities. Welcome, Noni. I think you might just be on mute.
Thank you very much, Watu, and thank you to the organizers for giving me this opportunity to share our experiences on engagement from Kenya, and especially focusing on the COVID-19 pandemic. So as of Friday last week, we were at over 100 million cases worldwide of confirmed COVID positive cases, and unfortunately, slightly over 2 million deaths as well. Within the continent, African continent, we were at 3.5 million cases and 88,000 deaths, with South Africa leading with 1.4 million cases and 43,000 deaths. Closer home in Kenya, we were at 100,000, slightly over 100,000 confirmed COVID positive cases and over 1,700 deaths. Now, the first confirmed case in our country was announced on the 12th of March. And thereafter, on a daily basis, we began to see an increase in infections. And this prompted the president to outline measures to the public that were aimed at mitigating or flattening the curve, as it was called. And some of these measures included a ban on international travel, and later restrictions of travel within the country, closure of all learning institutions, closure of places of worship, restrictions on a number of people gathering for weddings and funerals. And also staff were encouraged to work from home, both government and private institutions, while in other cases, staff were actually sent home. So while all this was happening, as you can see, there's somebody having a panic attack here. And there was a lot of worry, there was fear in the population. People were worried about what this meant for the future. And all of these, as they were happening, we were quite certain that this was going to affect engagement the way we knew it. So our community public and policy engagement approaches at the Kemri Wellcome Trust have been going on for over a decade now. Our strategy is grounded on action research, and the findings feed back directly into institutional policies, as well as making amendments to our engagement activities where needed, and we share our learnings widely. So who exactly are we? We are a large collaborative program, a collaboration between the government's Kenya Medical Research Institute, the University of Oxford, and the Wellcome Trust. And we have administrative offices in Kilifi, as you can see on the map, which is to the south of Kenya, Nairobi, which is the capital city, and Mbale, which is in eastern Uganda. We also have the largest HDSS in sub-Saharan Africa with a population of nearly 300,000 people. And the map just next to the map of Kenya is the map of Kilifi County, and the area that is shaded blue is the HDSS area. We also have a clinical information network of 20 hospitals, and our research is conducted locally within Kenya, the small orange dots, and the larger East Africa throughout the continent, and even international collaborations. We are slightly over 30 years old and have a staff capacity of 800. So our research activities are conducted in diverse contexts. Kilifi is primarily rural, but with rapid urbanization taking place in some of the towns. As I've said, Nairobi is our capital city, metro, a very cosmopolitan population. Mbale, a mix of urban, peri-urban, and rural. We are divided into 4 scientific departments: biosciences department, where we host our biobank, and we conduct basic science studies, immunology studies, clinical research department, from where we run clinical trials, health systems and research ethics department, from where we look at research on health systems responsiveness and empirical research work on complex ethical topics or complex research topics, and an epidemiology department, where we run surveillance and epidemiology type of studies. So as you can see, we are a large program running quite a large array of different types of studies. And so to support our research work, we have a very robust community, public and policy engagement strategy or platform, from where we have a diverse range of approaches that we use to engage the communities and the broader publics. I'm just going to give a snapshot because of the time limit here. So we have a CAB-like structure, a network of community representatives, and we engage this specific group of people regularly. We also engage primary and secondary school students, and these approaches and others enable us to have deliberative consultations with information giving that helps to foster understanding and co-learning between researchers and community members. Some outputs from the deliberative consultations that we normally do are, as I talked earlier, institutional policies. For example, out of a consultative forum on data sharing, we developed an institutional data sharing policy, and we also developed an institutional guideline on research benefits and payments that aids scientists. And we also publish a lot of our learnings. Another arm that we have is called study or project-specific engagement. And in this, we support researchers to think through the type of engagement that is needed for their studies, right from onset when they're thinking about the study and all the way to implementation and closure of research activities. So as you have seen in all these photos, our activities are very face-to-face physical activities where you actually need to meet. So the onset of COVID-19 brought immediate challenges. The first challenge was that our studies had to be stopped, and there was very little time between the time the president announced the measures to the time we were stopping. There was very little time to plan, and so we had to quickly put our heads together to think through how we were going to support study participants, especially those that were on investigational products, where they would go for support, how they would reach the study teams, how they would call, and so on and so forth. Focus shifted from our regular research work to COVID, supporting the government in COVID response. So we became a national and local testing center, and we also now moved into conducting COVID-19 research work. And nationally, at the national level, COVID was framed as a national security issue. So the challenge to us was it limited our involvement in information sharing. Now, I've put this slide here. It's a very busy slide, but what I'm trying to communicate with this slide is the complexity of engagement that we had to deal with and continue to deal with for our study, the COVID-19 vaccine trial. On the left part of the screen, we outlined the stakeholders that needed to be engaged, and then the corresponding colors show the different levels of stakeholders that needed to be engaged. So on the right, we have similar colors, meaning that the orange, for example, was engaging, for instance, the president, funders, National Assembly, and so on. And then the crisscrossing arrows generally show the communication lines. And then within our institution, we also had the shaded boxes. We had a team that was regularly communicating all the time, thinking through what are some of the things that need to be done and how do we move engagement forward. Some of the lessons that we're learning from this is that the principal investigator, for example, is very, very critical in engagement. They have to have a good understanding of engagement and even how it's going to be implemented. And that with all these crisscrossing lines of communication, there has to be really, really good coordination to ensure that we are not confusing our community members and stakeholders. And lastly, that we had to be really, really flexible because sometimes things came up and they made us go back to the drawing board in terms of thinking about the engagement. So other challenges that we face specific to our engagement activities is obviously as we went to work from home, our staff were removed from the community and the community felt the difference and they started asking, you know, where are we? Where did we go to? Why are we no longer in their households? Events were unfolding really quickly. We had to be thinking on our feet to ensure that there's no information gap amongst our staff, there's no information gap amongst the community members. Staff that went home, we were not able to provide everybody with equipment, mostly because of their own security. And we also had to think about, should staff then take mandatory leave? Those are some of the things that we were grappling with. So what kind of opportunities came our way? We became, we were more widely known. Of course, people knew about us as a research center, but with COVID-19, we were thrust out into the limelight. Our researchers were embedded into local task forces, both at the local level within the county in Kilifi and at the national level and other counties where they were providing technical support in terms of COVID-19 response. Due to our scientific expertise and the type of infrastructure that we have built over the years, we were able to conduct quick research. And this research was able to have the government get some feedback, some findings that enabled them to then make quick decisions on a daily basis related to responding to the pandemic. We also got pulled into or we got requested to join task forces, technical working groups where people were discussing how do we engage in this timeline where we need rapid research to be happening, but we don't want to leave community members behind. So for example, I and my colleagues are part of a WHO task force looking at good participatory practices for COVID-19 research. Other opportunities that came our way, for example, we have been running a radio program for about, I think about 3 years now. So we leveraged on these partnerships that we had developed over time, and we were able to get free airtime. Remember, we had shut down our face-to-face engagement activities, but we needed an avenue where our scientists could continue to talk to the community and continue to have conversation with members of the public. So we were able to get free airtime. The media houses and even more media houses gave us free airtime, and our scientists were able to go on air to talk about COVID-19 research work, and also just to address any other type of concerns that the members of the public had related to COVID-19. We became a source of credible speakers. So our media houses in Kenya, international media houses, sought us out to ask for experts to go to their programs to be able to talk about COVID-19. We got innovative. We started thinking about how else can we engage the public. We developed animations. And the picture you see down here with the coronavirus is a picture of an animation that was developed by one of our colleagues to actually together with the principal investigator of a vaccine trial. And it was an animation that we are now using to engage the public. And it was picked up by one of our leading media houses, and it was featured on primetime news. We moved some of our engagement to social media platforms, such as chat groups like WhatsApp. Like for our community representative group, we formed a WhatsApp group with the community representatives who had smartphones. And so we began to engage them using that platform. And then we also used Skype for Business to have meetings via mobile phones with this group of people. And while all of this has been happening, we decided to come up with a document where we would be writing down the decisions right from the time the first case was announced, all through the different phases that we passed through, the decisions that needed to be made for the research activities, for engagement activities, the dilemmas that were facing us, and we were able to be discussing all of this. This is very massive work. As you have seen, we are a large research institution. We conduct a massive amounts of research work, and I don't do it alone. So kudos to this really wonderful team of experts that is very supportive in our work. Thank you very much.
Well, Noni, thank you. That talk, there was a lot packed in there and very clearly presented. And of course, the takeaway message there is communication, and very well coordinated and aligned with military precision to deal with the emergency. I mean, it's impressive how so many stakeholders came on board to help that happen. Now, please hold your questions for Noni. We'll come back to her in a little while. Our next talk is by Olu. So now over to you, Olu. Olu is going to speak on developing capacity for Africa's genome revolution.
Thank you very much. Can you hear me, Watu?
Yes, I can hear you very well.
Thank you very much, Watu, for that very generous introduction. So I would like to start this talk by introducing you to a man who once fed the world. So the elderly man you can see here in between Nancy Pelosi and the then President Bush is Norman Borlaug. And you could be forgiven to think that he was a politician. No, he wasn't a politician. He was a scientist, and actually he was a plant breeder. And after World War II, the global population started increasing drastically. And there were fears of famine in places like India and Pakistan. So what Norman did was that he led a movement that is now widely known as the Green Revolution, in which he developed scientific innovations. Particularly, he developed improved wheat lines. He trained local researchers, and he worked a lot with government. And at the end of all of these, places like India and Pakistan not only were able to feed themselves, but they became net exporters of food to other countries. Now, it is estimated that Norman fed about 1 billion people in the world. And because of that, he won the Nobel Peace Prize in 1970, and he was given the Presidential Congressional Gold Medal. Now, I want to argue that Norman's experience is very relevant for us today, even in Africa. And the reason is because Africa's population is growing. So this graph here, it shows the trend of the world population. And you can see particularly that Asia represents a large chunk of that population. But I want to focus your attention particularly to the dotted line that tracks the population of Africa. And you will see that in the coming decades, Africa's population is growing, and Africa is expected to become the new Asia. So because of this growing population, my talk today is going to focus on what are the opportunities that are becoming available for us as African researchers, or even those who are not researchers, to collectively usher in the green revolution, just like Norman Borlaug did. As a geneticist, I'm going to focus my talk mostly on the genetics aspect of it. And I will show you a few examples of some of the research I've been privileged to be a part of. Now, given the broad nature of this audience, I will not go too detailed into some scientific results, but I thought it would be a good start to give you a crash course on genetics. So I will just introduce you to Genetics 101. So many of you are perhaps out of DNA, and you might be thinking, what actually is DNA? So think of DNA as a very long string that is tightly packed within the cells of living organisms. And those living organisms could be plants, could be animal, or even human. But importantly, there are parts of that strings that contain information, or specifically instructions that directs the activities that take place within those living organisms. And those parts is what we call genes. So those instructions are in the form of chemical codes that are mostly made up of four substances, which are these red, blue, purple, and yellow substances that are arranged in a particular format. Now, we usually call them G, C, A, and T just as an abbreviation of their names. Now, the best way to describe this is if you think about this as a book or like an instruction manual that is written in English and contains English sentences. And within that book, just like you will find uh, alphabets within this book, English alphabets like A, B, C, and D's, those represent the nucleotides. And you could arrange those alphabets in certain orders to form words. And those words can form sentences, like I love to see the smiling faces of farmers. And those sentences represent the genes because it's carrying an information. And within English, sometimes spelling mistakes can change the meaning of a sentence. For example, just adding an additional E to the sentence completely changes the meaning of the sentence. And in the same way, uh, changes in DNA, which we call mutation, can change the function of a gene. So let me illustrate this to you with the story of another elderly person that I met, and hopefully to make it clearer. I was really privileged to meet this woman. She's Wendy Karoya. She was 82-year-old old when I met her. And it was a privilege to stay with her in an Airbnb accommodation in Kenya. And this was really important for me because I just moved, returned back to Africa from the UK, and I wasn't feeling too confident about my decision. So I was having a double mind. But this experience really was crucial for me. So when she learned that I work on wheat, she went into a room and brought these two jars of wheat to me. And these are wheat jars that she's kept for 60 years. And they're from a particular wheat varieties that the one her husband, the best wheat grown in Kenya in 1960. Encouraged by the success of that year, they decided to grow the same wheat variety the next year. But unfortunately, they lost almost all of that wheat. And Wendy did not know that the problem she was experiencing then was the focus of my PhD research, which was why I was so excited about it. So the problem she was experiencing there is a problem called pre-harvest sprouting. And as you can see, these are cases where grains start to germinate even before they are harvested, especially when there is rain. But varieties have genetic resistance to this. So there are some varieties that are resistant to this problem. For example, in this image, so these spikes were from two different varieties, but they were grown under the same condition. And you can see one is quite resistant and the other is not. So for my PhD work, I was fortunate to contribute to a research where we identified the gene that is actually responsible for this difference. And just like I showed you previously, how a spelling mistake can completely change the meaning of a sentence, what we discover is that just a small change in this gene, just from a C to A, is what is partly responsible for this effect that we see. So this further illustrates how the changes in the DNA can show effect on the outside. And spurred by my discussion with Windy, when I came back to Africa, I decided to take as many wheat varieties or the wheat varieties grown in East Africa for the last 100 years and just ask the question, do we have the good version or the bad version of these genes? So the red bar in this graph represents the bad version, the frequency of the bad version. And the blue bar represents the frequency of the good version. And as you can see, over time, we've been accumulating and growing more of the varieties that have the good version. So in this particular case, since when this time, things have improved a lot. But we can't say the same for all of the other traits that we want in our crop. And I will show you a very important example. So plants also have pandemics, just like human beings have pandemics. And there was a particular pandemic that happened about 2 decades ago in wheat, which is called the UG99 pandemic. And just like the COVID pandemic started in Wuhan, China, unfortunately, in this case, this particular pandemic started in Africa, particularly in Uganda, and that was why it was called UG99. And it's so important because it threatened wheat production globally, not just in Africa, but globally. Now, we have tried a lot and researchers have tried across the world to develop lines that are resistant to this problem. But the problem is that the pathogen keeps changing and it keeps bringing a very aggressive form. Just like in COVID-19, you have new strains and new mutation, the UK mutation and the South African mutation. So the pathogen is changing and is giving very aggressive form. But thankfully, there are some genes, like one gene called SR26, that confers resistance to all known forms of these pathogens. Unfortunately, in Africa, we don't have these genes. We really don't have these genes at all in Africa. So this highlights one of the examples of opportunities in which genetic improvement can improve some of the things of the crops that we grow in Africa. And therein lies the focus of my research. So in my research, what we try to do is to develop better yielding and nutritious crops, particularly wheat. And the way we go about this is by trying to combine and introduce genes that are beneficial, but that are missing in Africa. And we try to combine them and introduce them together in a common wheat background using conventional breeding method. In this particular case, we are not using GMO technology. And it takes a long time, and it typically takes about 10 years or more to do something like this. And so what we are doing is that we're using an approach that is inspired by scientists from NASA that they use to try to grow plants in space. And it just mainly entails growing plants under constant light for, instead of 12 hours a day, you can grow them under constant light for 22 hours a day. And you can see the plants here, the two plants here, they were grown at the same time, but one was grown under speed breeding condition and the other under normal condition. And you can see that the plant on the left is grown much faster and is maturing much faster. And this, we hope, would help us to accelerate this step so that we go through all of this generation in quick time and we don't have to wait for 10 years to achieve this. And I'm really fortunate because we've been able to manage, we've managed to combine some of these genes together with the help of these amazing scientists that I show here. And we're just waiting to test the effect of the genes in the field, hopefully this year. Now, so this is just one example out of many examples in which, by understanding the genetic makeup of a plant, you can use that information to improve the crop. And this is not just applicable to wheat, it can be applied for many crops in Africa. And this is important because Africa is blessed with a lot of diversity. And not just in terms of crops or livestock, but even in terms of humans. So it is believed that Africa has the greatest genetic diversity in the world. But the problem is this, that although we have this genetic diversity, many of these crops that we have are under-researched and they are underutilized. And what I mean by that is that for many of these crops, we don't even know what their genomes look like. Just like I showed you the open book to explain what genome is, where you can read the sentences in this book. For many of the crops that we have in Africa, it's like a closed book. It's not that they don't have a genome, it's just that we've not been able to read their genome. We've not been able to sequence their genome to find out what genetic information they have. And if you can't do that, you can't identify genes that will help you to improve certain traits in a crop. And the reason for this is because we lack the infrastructural capacity and the human capacity historically in Africa to do this. To illustrate this, let me just show you. So these are some of the machines that are typically used to decode the genomes of crops or plants or livestock. And you can see that they're usually big machines, they're expensive, and many researchers in Africa cannot afford this. But there are new opportunities that are now becoming available to us that we can take advantage of and which we have been taking advantage of in my research. One of them is a new technology called the Oxford Nanopore sequencing technology. Interestingly, it was developed around Oxford University. So I think it's aptly fit for this talk. And it's cost just £1,000, $1,000, and you can hold it in the palm of your hand. And with something as small as this, you can start sequencing the genomes of crops, of animals, and even humans. And some people have used this in Africa to sequence the Ebola virus and even the COVID-19 virus. Now, this field is not just about machines. So you could have the machine you want, but you also need people to do this. And historically, Africa has not taken a front seat in most of this effort because of the cost implication. So historically, most of the sequencing has been done by people in the global north. And we really appreciate that because we want people all across the world to support research in Africa. But we also want African researchers to take the front seat in many of these efforts. So I'm really fortunate to be part of a team where we identify this gap that most of the sequencing is just done outside of Africa when it comes to really important sequencing. And we formed a training initiative where we gathered 14 African researchers together from 8 African countries. And we noticed that most of the training that have been around are usually short, 1 week or 2 weeks training, which are really introductory, but they don't really consolidate the skills that you need. So we decided to take a different approach. So we brought all of these researchers together in Kenya, and we trained them here for 8 months on different aspects of genomics and bioinformatics and on full scholarships. And with this success in our training, we got the encouragement to start taking big and ambitious projects. And proud to tell you that with this training, we started taking on some genome sequencing projects. And these are some of the genome sequencing that we are doing on some of these orphan crops. Crops like teff, like Lablab, and African yam bean. And we're privileged to collaborate with some of these people on this crop. For Lablab and African yam bean, we've been able to sequence the genomes of this crop. And most of these, particularly for African yam bean, was done in Africa. It's something that we are proud of, that we also can actually start sequencing the genome of crop. Something like teff has been sequenced before, but we are starting a new initiative to resequence more varieties of teff and be able to decode the genetic information within this. So these are exciting times because of the nature of this talk. I won't go into the scientific result, but we are getting some interesting result. And more importantly is that we're developing capacity for us to be able to decode these genomes. So I would like to summarize my talk by just saying that we need a green revolution that needs to cut across both staple and orphan crops. So I've shown you an example for wheat, where understanding how a gene function, you can use that to improve certain crops. But we also need to look at orphan crops. And in most cases, these orphan crops are under research and we don't even know about their genomes. So sequencing these orphan crops genome is actually the next frontiers of genomic advancement. And I think the most important message for me here is that, particularly for orphan crop, when you sequence a genome, it's like you're giving birth to a child. And I think the parents of the child should be there in that vetting experience. So I think African researchers should be an integral part of all of these genome sequencing efforts we are doing, particularly for African crops. Lastly, I would just like to thank some of the people who fund this work. So my work is mainly funded by the Royal Society through the Flair Fellowship. And the training we've been doing has been funded by the BPSRC. And I'm really fortunate to collaborate with all of these institutions. So thank you very much, and I'll be happy to take questions.
Thank you, Olu. Another fantastic talk. I think this evening we've had a very real treat. And, plants also have pandemics. That's a very captivating way of putting it because, obviously the concept of pandemic has become very real to us and everybody understands that. And also the many parallels you've drawn between crops and human health, that not just clinical trials can be sped up, so can crop breeding, for instance. And then, the very extensive diversity of these African crops and, vis-a-vis the well-known genetic diversity amongst African populations. Now, we have a number of questions, and I'll start with one for Noni that has appeared very many times, and I'm sure you are expecting it. So please take your time to really give us as much information as you can on this. So the question is about vaccine trials. So the vaccine trial was a source of a lot of community anxiety. How did your team navigate the publicity, the negative publicity?
Thank you, Watu. And I don't know who has asked the question, but thanks for that question. You're right. Right from the time, even before we started conducting the vaccine trial, when we were still thinking about it, we had the case of the two French doctors who, were filmed, saying certain things about research and Africans. So already that created, some small chaos on perhaps small, perhaps large, on social media, and people were already, having their antennas up in terms of vaccine trials. The way we navigated this and the questions that up to now still continue to come to us is generally what we do: engage the public, give them information. So on that very busy slide that I showed, we started our engagement way early on, and we demarcated, or we organized ourselves into teams: who is going to be speaking to the Ministry of Health, who is going to be engaging the Cabinet Secretary, who is going to be engaging the local Department of Health here in Kilifi, and who is going to be engaging the broader public. And so we started our engagement way early on, talking about clinical trials in general, to increase people's understanding, including the public. We went on radio. We had a number of our scientists going on radio to initially just talk about clinical trials. What are drug trials? What are these trials where somebody is given an investigational product? How safe is it? What happens in the lab? How do they start? So we tried to demystify and respond to all those questions. And eventually, obviously, we also got to the COVID-19 vaccine trial itself. And we had mapped out, as I showed you, we had the animation that shows very clearly how the vaccine has been made. And basically, we are still soldiering on because the questions are still there up to now, and anxieties are still there.
Okay. I'll give Olu a bit more time to catch his breath. So I'll follow on with another question for you, Noni. What has been the most important lesson for engaging the public in the pandemic? And do you think these new adaptive approaches, because very clearly there was a lot of adaptation and a lot of coordination that you presented, are the future of engagement?
Thanks for that. I don't know if I have one main lesson. Perhaps I have several lessons. If I was to speak about one main lesson, is that people just want to know. And you can't keep information to yourself. And when there's an information gap, or when there's only one voice that is talking to people, then that gives rise to rumors and misinformation. So people just want to know. People just want to be involved. They want to be to have their voices heard and to also voice their opinions and know that their opinions are listened to and their opinions are taken seriously, and somebody is going to act on things that they are expressing. So that was, I think, for me, one of the really, really main lessons. If I can say another one, is that the situations are always fluid. You can't make your plan and cast it on stone. You must be adaptive. You must be able to go back. If something comes up, you must be able to go back to the drawing board, and be able to, sort of like look into how differently you can do things. The new approaches that we are using and whether that's the new way to engage, certainly there are some things that we are going to carry forward. Carrying out virtual meetings is something that we are going to continue doing to the extent possible. However, it does not entirely rule out going back to some face-to-face activities, because all of us as human beings, we value the human touch. We value actual interaction. There's something about sitting opposite someone who is talking to you, seeing their face and being able to actually, understand through their body language and everything. So we will go back to some face-to-face engagement.
All right, thanks. So Olu, I have two technical questions for you that I'll ask, and you might be able to respond to them together. So it's about the gadget that you showed us, and one person has observed that the wheat genome is much more complicated than the mammalian genomes, and how does this gadget cope with that? And the second question is about the availability of cheaper sequencing platform. And then, so the person is wondering, the interpretation of results in addition to other bioinformatics expertise is a real challenge in our setting. Could you please speak to those two questions?
Yeah, thanks, Watson. Those are really good questions. So the sequencer that I showed you, the small one, it's we mainly use it for sequencing orphan crops. So the Lablab and the African yam bean that I showed you, they have smaller genomes, and we've used them to sequence those ones. Yes, it's true. With the wheat genome, it's really massive. So it's about 5 times the size of human genome. Although you can use the sequencer to sequence some parts of the wheat genome through some algorithm that selects certain parts that you want, selectively select, pick certain part that you want, but you don't necessarily use it to sequence the full genomes. For that, you have to go to the bigger platform. But for many crops in Africa, you can conveniently use the small device to sequence those crops, and not just crops, even pathogens as well. As I said, some people have used this for Ebola and for COVID. And the second question about capacity building, it's really important. And that was why I showed a slide that it's not just about sequencing these materials. What matters is how do you analyze the results? Because when you do the sequencing, you get a lot of data, you get big data, and then you stock, you get, so what typically happens now is that African scientists have woken up to the importance of sequencing. So what they do is that they engage people in the West to do the sequencing for them, and then they get this data back, and then they don't know what to do with it. And so the training we did wasn't just about the sequencing. The training we did was to train people on how to analyze the data. And so the sequences we generated for those orphan crops were actually analyzed by the fellows that we train. And I think that's the model that we want to carry forward. So that's the really important point there.
Yeah. And there's some interest in Wendy from Mombasa. Maybe you can tell us more about Wendy. How many examples do we have of older wheat strains that have been used on the continent?
There's a little bit of history. The problem is we don't have proper documentations. And historically, people have thought of wheat as a colonial crop and not as an African crop. And although that was true, because it was introduced to Africa by the colonial master. But that story has changed a lot. I couldn't show one slide where it shows that actually Africa has taken up a lot of wheat now. And I could ask the people on this platform when was the last time they ate bread, and it could be either early this morning or yesterday, because we are eating a lot of wheat now. But historically, we've not documented some of the important role that wheat plays in Africa. So that's a counter I had with Windy was a very chance encounter in Kilifi, where Noni is based. And I wish I could spend more time going about and looking for more stories like that.
Okay, thank you. You gave us a primer, a very useful primer on crop breeding, but Noni also did a very good job on bringing us up to speed on community engagement. So I'm sure you'll be able to take this question, all that's about engagement. So if we link your talk and Noni's talk on community engagement, how do plant breeders engage with farmers? And are there new exciting methods of engaging in the same way we've seen Noni describe the situation in global health?
Yeah, I think I need to learn from Noni on this, because plant breeders have historically been confined to the labs or to the field. But typically what we do sometimes is we have open days where you grow out the materials that you've developed, and you invite farmers to come and have a look at it and evaluate it. The one important point I should make was for Norman Borlaug, the guy I showed you earlier. I think what really helped his work was his engagement with the community, and not just with the community, with policymakers. I showed you how he met with several presidents, and he went across Asia meeting with several presidents and just talking to them about his work. So he understood the power of going out and talking to people about your work. And I think the important thing there is, just like Noni talked about, sharing the correct story or showing them multimedia to illustrate the story. When people see things for themselves, they start to believe it. When you show them that wheat variety and how it is better than the other one, they will start to believe you. And so that's one way we've been doing community engagement, mostly by demonstrations. I don't know, maybe Noni has something to share with us in that.
Yes, I was going to invite her to tell us, give us some suggestions on how farmers can better engage. If there are any lessons to be drawn from your area.
Yeah, I mean, I think the methods of engaging the community are diverse, and they are similar across very different types of interventions and programs. So, for example, we borrow quite a bit from the development world. So, as you said, Olu, you have open days. We also have open days. We invite community members into our campus. They visit our laboratories. They sit in our seminar rooms, and scientists come into the seminar rooms to present their work. And they are sometimes asked like really very tough questions by community members. So we do those kinds of things. I talked about radio. Recently, we have been piloting out using drama, community drama, to talk about health research. Because of COVID, obviously, now we have had to move that community drama onto radio, but it is still the same, working with a local theater group, training them to understand what health research is, so that when they are scripting, they're able. So for farmers and for plant breeders, it's taking everyday life and then being able to develop something that can communicate or can spark a dialogue, or can incite people to start talking using whatever means, an animation, a drama, a short film. There are many methods.
Fantastic.
Yeah.
A very practical question for Olu. In all the research you've been doing, are you able to tell us a little bit about gluten and gluten intolerance? Are you on the way to coming up with a breed of wheat that is less gluten, has got less gluten?
So there are two answers to that. So the short answer is not yet, because it's a very complex problem. So when you talk about gluten, you're not just talking about one gene, you're talking about a class of genes. So you're talking about many genes, and some of them are close to each other, others are widespread. And there have been efforts, not me particularly, but there have been efforts globally to get rid of some of these genes. But it's a catch-22 situation, because to make good bread, you need the gluten. So if you don't have gluten, you can't really make very good bread. The quality of the bread will not be good. But you can still make bread without gluten. It's just that the quality wouldn't be great. So the short answer to that is we are not yet there yet, but we're working towards that. The other answer is that there are many alternatives to wheat for making bread, like the teff I showed you, or there are other cereals that you can use to make bread that are absolutely gluten-free. So yeah, maybe in the next 5 years or more, we might see some things in the market in that direction.
And just to follow up on that, like the teff you're mentioning, why aren't these crops more popular?
So teff is, yeah, I didn't mention that during my talk. So teff is more popular in Ethiopia and Eritrea. So teff is actually very unique because it's one of those orphan crop that is so central to food security in certain parts of the world, like maybe in Kenya or Nigeria, where maize and rice is king. In Ethiopia and Eritrea, teff is king. So morning, afternoon, night, you just have teff. And if you've been to an Ethiopian restaurant, you would be familiar with injera. So it's a flat bread that people eat, and they eat with different sauces. And that is what teff is used for making. For the African yam bean is quite popular in West Africa, some part of East Africa, but Lablab, I think Kenyans call it injai or something like that, or black beans. And I love it. I eat it whenever I have the opportunity to. So yeah, I think it's widespread across Africa.
It's traditionally supposed to be very nutritious and very highly recommended for mothers with newborns, actually, and
Yeah.
good care, as I understand it.
Yeah. I'm not pregnant, but I still eat it though.
All right. So Noni, I was just wondering about, obviously you work at Cambry and so on, and in some instances there might be situations where you might be wanting to communicate a specific message that is totally unbiased or opposition and so on. How are you able to strike a balance between that message and being viewed as representing a certain, say, research center? Can that sometimes get in the way of the message? Are you on mute? Yeah, you're on mute.
I was saying, just ask that again. How do we strike a balance?
Yeah, so it's about striking a balance between the unbiased position you might be wanting to communicate and perhaps some historical sort of misalignment or misunderstanding about the institution that the person is coming from. So when you try to give an unbalanced position, all people are seeing is the institution you're coming from, and then struggling to sort of embrace the unbiased message.
Thank you for that, Watu. So what we have tried to ensure that we do within our population specifically, and even just within the country, is to just be honest, to be an open book, and just tell things. I can actually say almost as they are. So, right, I could be speaking to a group of people who perhaps have faced issues or have issues, and we do have sometimes community members who feel as if the institution has either harmed them. They have perceptions that the institution has caused them some harm. So we have we have an interesting system. We have a very specific system that we have set up where we try and approach the people and speak to them ourselves, if at all they will listen. If not, we also try to work very closely, for example, with the Department of Health in Kilifi, that is seen as they're not favoring anyone, or they're not on the side of anyone. And we go together with them to community meetings, for example, and they're able to either back us or give the actual position of whatever information it is that we are giving. We try to partner a lot so that it's not only just us who are engaging with the community about the topics that we want to engage in, for example, research. So for instance, community members would say, well, you've come to talk to us because you need to recruit us. Maybe what you're doing is not so important. But when we have a Department of Health staff accompanying us and also weighing down that message that research is important, then it removes that bias from our part.
Fantastic. Right. I'll just quickly check whether you might, both of you or either of you might have anything else you'd like to add that maybe you didn't get a chance to say that you thought was important, but in perhaps just 1 minute, if you may.
I think I was able to say everything that I wanted to say. So, Olu, maybe. Olu, you're muted.
It's been a privilege to give this presentation, and hopefully there will be more opportunities for us to engage with people and share more about our research and maybe talk about some results that we're getting. So thank you for this opportunity. Yeah.
And Olu, how are we doing on the front of capacity building across Africa in your area? I mean, are we moving in leaps and bounds? I mean, I know in global health, there's quite a lot of progress, but how do you see it in your area?
I think for crops particularly, we are not there yet. When it comes to human health, I think Africa is progressing. Like there are initiatives like the H3A Africa, where a lot of capacity building has been done in that area. But for crops, it's almost like neglected in a sense, and that is one of the reasons why I'm here, and one of the things I want to commit to see how we can raise the crop profile for building capacity for genomic research in crops. So if you look at most of the publication around crop sequencing, you will see that it's dominated by people from the global north, and you find African researchers mainly collecting samples that they send abroad. We want to change that narrative, and we're hoping that maybe efforts like these can start to gather momentum, and we can use that to change the narratives for crop research.
Thank you. So it remains for us to thank the speakers and the audience, but before I let you go, I'd like to let you know that we have some great news. AfOx has a new series of talks on decolonizing archaeology. And the first one is going to take place on 19th of February. We have all this information on our Eventbrite page, and I think a link has already been shared on that as well. And then I'd also like to let you know that our next insaka will be at the same time, that is 5 p.m. GMT on Thursday, 4th of March. And we have two great speakers, two women, one from Uganda and a second one from Nigeria. And we hope to see you then. So thank you, everybody, and we shall look forward to seeing you next month.
Right, the live stream has stopped.
Oh, okay. What happened to Avni, the producer?
Avni got chucked off Zoom, but we're still online, strangely. So she called me on WhatsApp. She's going to try and rejoin. So I had you on speakerphone as you were ending with her with the button ready to end the live stream.
Okay, all right. Maura, did we share the wrong